Hello, let's discuss on tissue culture. Tissue culture is a technique of growing plant cells, tissues or organs in a sterilized nutrient medium under controlled environmental conditions. The plant, the plant parts or the plant material that is to be used for tissue culture are called as the explant, and this explant can be excised from root, shoots, flowers, enter or from any other part of the plant. Now, when this explant is grown in a nutrient medium, this explant they begin to divide and develops into whole plant body again. So this capability or capacity of the explant to grow and develop into whole new plant body is called as totipotency or cellular totipotency. Now, we shall be discussing on the procedure of tissue culture. The tissue culture involves following steps. The first step is sterilization of explant. The plant tissue that is to be used for tissue culture is excised from the plant from its original location. The second is sterilization. In this stage, the culture media, the culture vessels, the instruments to be used, and even the explant are made free from microbes. So this method is called as sterilization. In case of explant, the explant is treated with specific antimicrobial chemicals such as chlorine water, sodium, or even calcium hypochlorite solution. This treatment is called as the surface sterilization, and in case of culture vessels, culture media, and the instruments, the sterilization is done with the help of steam or by autoclaving the required materials for tissue culture. The third step is the transfer of explant to the culture medium. Once the explant, the culture media, and all the uh, required materials are being sterilized, the explant is introduced into the sterile culture medium under aseptic condition. Aseptic refers to the condition where it is free from microbes. So under aseptic condition, the explant is introduced into the culture medium either in solid or liquid culture medium, and this setup is kept under aseptic condition with optimum growth with optimum condition for growth. The fourth stage is the callus formation. Once this explant is introduced into the culture medium, the explant they begin to undergo divisions, and within around 2 to 3 weeks of the culturing, this explant begin to develop a mass of cell called as the callus. So this callus is an undifferentiated mass of cell that arises from the explant. Now the fifth is the organogenesis and formation of plantlets. This callus, under the influence of various concentration of different types of growth regulators, the callus undergo differentiation and it begin to develop shoots, roots, and give rise to miniature plantlets while it is still in the nutrient medium. The growing tissues depend upon the sources of selected tissues, that is the explant, condition of growth, and even the composition of the nutrient medium. So this is all about the procedure of tissue culture. Now we shall be discussing on the types of tissue culture. The first is the callus culture. In callus culture, the explant is grown in the nutrient medium that is containing agar gel. The explant, after about 2 to 3 weeks of its introduction into the nutrient medium, begin to divide repeatedly and begin to form an unorganized mass of cell called as the callus. So the callus remain on this medium suspended, and this nutrient medium is being fortified with growth regulators like auxin and cytokinins. So these are the growth regulators that is being added to the nutrient medium. Later on, the callus that is suspending in the nutrient medium differentiate and begin to develop a new plant body again. So this is all about callus culture. The second type of tissue culture is the suspension culture. In this type of culture, single cells or group or small group of cells are suspended in the liquid growth medium that is containing growth regulators such as auxin, and there is a continuous agitation of this culture medium at around 100 to 200 round per minute. This agitation of nutrient medium help to provide constrained mixing of medium and also help in breaking cell aggregates and aeration of the nutrient medium. As a result of this, the suspension culture, the X plant begin to grow faster as compared to the glass culture. Therefore, the suspension culture is more efficient than the glass culture. Now we shall discuss on subculturing. Tissue culture cannot be maintained for an indefinite period of time because the biomass of the cell begin to increase considerably. Even the level of the nutrients in the culture medium begin to decrease, and the volume of the culture medium decline due to continuous use and its evaporation. Therefore, some cells or piece of glass from the main culture have to be transferred regularly into new culture vessels containing fresh media. So this process of transferring some parts of the cell or tissues from the original culture medium to a new culture media is referred to as subculturing.